Hello everyone, hope you all are fine and doing well. Here is Dr. Hira Bashir, MPhil Pharmaceutical Chemistry. Today we are going to discuss about microbiology of air, water and soil. As we know, microbiology is basically the study of microorganisms, the study of small living creatures in our environment. And that environment may be air, may be water, may be soil. We will discuss which type of microorganisms are present in air, which type of microorganisms are present in water, and which type of microorganisms are present in soil. Microbiology of air, first of all we will discuss here. What is air? Atmosphere, basically. According to World Health Organization, the air is basically, air pollution is a situation which are harmful to people or the health. Any situation that is harmful to the people or harmful to their health is basically air pollution. According to University of British Columbia, the term air quality means the state of air around us. The state of air around us. Good quality air refers to clean, clear and unpolluted air. Alright? If the air is of good quality, it means it is clean, it is clear, it is unpolluted. Pollution not only means that it contains smoke, it contains dust, it contains moisture. Basically, the pollution means here what we are discussing is the pollution refers to the microorganisms which are dangerous to human health. The clean air is essential to maintain the delicate balance of life on the planet. If the air is clean, it is very essential, it is very necessary to maintain the delicate balance, which is very important in our environment. So the air must be of a good quality. Alright. First of all, air pollution, which is basically the situation which is harmful basically to the people. And according to British Columbia University, the air quality means the state of the air around us. If the air is of good quality, it means it is clean, it is clear, it is unpolluted. And it maintains our environmental balance. Environmental air, basically, we have different laws related to this. In Pakistan, environment related law in Pakistan is present. Pakistan Environmental Protection Act 1997 is present. And according to this Protection Act, it provides for protection, conservation, rehabilitation, improvement of the environment, for prevention and control of pollution, and promotion of sustained, maintained development. Alright? If we study different environmental protection acts, then according to Pakistan Environmental Protection Act, which was given in 1997, this act is to provide protection, to conservation, rehabilitation and improvement of the environment. For the prevention and control of pollution in the air and promotion of sustainable development. These 6 to 7 points are the major point of discussion in this act. Protection, conservation, rehabilitation and improvement of environment. The first thing. The second thing is for prevention and the control of pollution and promoting the sustainable development. In Indian Air Act 1981, ambient air pollution considered in legal sense for punishment. If the air pollution is present according to Indian Act, then that person or that thing is responsible for punishment. Whatever the reason for that air pollution is, that person is responsible for a proper punishment according to the Act. Assessment of microorganism in air, the major point of discussion. How we can assess, how we can identify, how we can check the microorganisms in the air. There are several methods to count the microorganisms in air. This is basically called assessment. There are several methods to count the microorganisms in air. The very first is the impingement in liquids. Impingement means to add, to absorb, to penetrate in liquids. Means we will adopt a method which will absorb, which will penetrate microorganisms into that liquid. Or the way we use, we can clear it from this. In this method, the air is drawn by capillary tube. We use a capillary tube, a very small tube, long tube, having small holes, both points are open. Air is drawn into the capillary tube through the fluid in the form of bubble. We have bubbles in that tube filled with liquid. Means there is a capillary tube, some liquid, then a hole, means a bubble, air bubble, then we have a liquid, then we have a bubble. Alright, we are discussing, keep in mind about microorganisms in the air. That's why we have a hole, we have a bubble in that capillary tube along with liquid. We are discussing about impingement in liquids, means we have to penetrate that microorganism in the liquid, and that microorganism present in air. We trapped small air bubble into that capillary tube along having a liquid. The organisms get trapped into the liquid. The liquids are covered with medium to determine its microbial content. We place that liquid in a media which is suitable for that growth. Living cells can be enumerated in this method. If they are living, if they have artificial media sensitivity, then they will grow. They will increase their number and we can enumerate them very easily. We can identify them very easily. Alright. This is basically impingement. We will have a small capillary tube. We have liquid in it along with some bubbles, and the air present in that bubbles contain microorganisms, and that microorganisms will be absorbed, will be trapped into the liquid, and that liquid is then placed in a specific media, and the microorganisms, the living systems were then enumerated. A very simple method. The other one, impingement in solids. We can penetrate them, microorganisms in liquid, and we can penetrate them in solids. How we penetrate in solids? The microorganisms collected and impinged directly on the solid surface of agar media by gravitational force. We have a specific media having a nutrition for the growth of microorganism. Alright? That media surface, the upper surface of that media is exposed to the air. Exposed to the air. As the media surface is directly in contact with the air, the microorganisms will get nutrition from that surface of media and start to grow. Colonies develop on the surface of media after a few days of incubation. Then after incubation of that media, providing specific temperature, specific humidity, specific environment for a few days, we will see that these colonies under microscope and we enumerate them. Cover the petri dish containing agar media is removed and sugar and the agar surface is exposed to the air samples. Petri dishes are covered having agar media. Then when we start our procedure, we will have a media which is covered, prepared is very specifically according to the method. Then remove the cover. Agar surface is exposed to the air sample. Colonies will develop, and technique does not record actual volume but approximate estimate. Because all microorganisms cannot be checked by using this method. As we have discussed that viruses cannot use that artificial media for growth. So we can only have some microorganisms like fungi or fungal spores if present in air and bacteria if present in air. Then we can have their development in the form of colonies under microscope. Or maybe somehow they can be visible because we have to draw a pattern specifically where we see these colonies. It gives kind and number of microorganisms in a particular area. Specific area have specific type of microorganisms grown there. A very simple method. The first one impingement in liquid and second one impingement in solids. Then source of contamination. How air is get contaminated? The basic reason is automobile, which uses fuel. When fuel combusts, pollute the air, releasing carbon in air. Agriculture source. When we spray different pests, killing sprays are present. These sprays damages our environmental quality, our air quality. Industries, especially tanning industry, where animal skin is converted into leather. That process pollutes air, releasing chemicals in the air, releasing gases in the air. Various nuclear power plants, they are very, very damaging to our environment. If they are present in industrial area, there will be less pollution in that area, but that will be less dangerous because that is not an area which is residential. So we plant these various points such as these premises like industries or nuclear power. Then we plant these premises in the specific commercial area, not in the residential area, to protect the people health. Chemical industry releases chemical in the air, very dangerous to the health. And which type of air pollutants are present in air? As you are seeing in this diagram, there is an industry releasing waste in air, and the air is totally polluted, but we don't know the nature of this pollution. Which things it contain, which chemical it contain, which gas it contain, which metal it contain. So these are basically the pollutants. Maybe carbon monoxide, maybe a lead, maybe nitrogen oxide, maybe ozone, maybe any matter, any particulate matter, sulfur dioxide, acrolein, asbestos, carbon disulfide, benzene, kerosene. Any of these are very few micropollutants. These are very, very few pollutants. Alright. Measurement of air quality. How we measure air quality? Air quality index is used. AQI is used to measure the air quality. The value ranges from 0 to 300. The higher the value of AQI, the greater will be the level of air pollution and greater the health concern. As it increases from 300 above, the air is totally polluted and it is very, very dangerous. But if the value is between 0 to 300, we can clear that. We can have chances to clear that air pollution. But if the values goes above 300, then it is very hazardous, very dangerous. Alright, for example, an AQI value of 50 or below represents a good air quality. If the AQ value is 50 or below 50, then the air is of very good quality. Above that value, convert this air into a polluted air. Alright. You can see here air quality management cycle. According to scientific research, it firstly the determination of emission reductions, the presence of industries, you can see here, is very far from the residential area. Various control strategies were applied to protect the air pollution. Various programs are implemented. Then evaluation is checked and the established goal is achieved. That is a proper and good human health. Then microbiology of water. Now what is water? We will discuss different types of water. And you will be very wonder that we don't know about that. We just see water and we can say it is simply a groundwater. But now we will study about different types of water, different layers of water, and the presence of living creatures in that different layers. Then water microbiology is defined as, we can say, aquatic microbiology. It is concerned with the microorganisms that live in water and can be transported from one habitat to the another by water. Those living creatures which are transported from one to other environment by water is basic discussion. There are two types of water, groundwater and surface water. Now what is groundwater? As the word indicates, deep water. It originates from deep wells and subterranean springs. Various springs, having waterfalls, deep wells. That water is deep water. That is free from bacteria due to deep sand and rock or filterable action of water. The presence of sand and rocks filter this deep water and make it bacteria free. It only gets contaminated when it flows from various channels. That from the surface, when it comes into the surface, then it flows from different channels of rocks, different mountains, then it gets polluted. But if it is coming from the wells, it is pure, free from bacteria. After getting in touch with different surfaces, that is polluted. What is surface water? It is found in streams, found in lakes, shallow water, surface water on the upper surface, upper layer. Contamination. Air contaminates the rainwater. The air contaminates rainwater, agriculture products, industries, waste and sewage. Air is basically responsible for this water contamination. They carry a lie in human intestinal tract like E. coli, Salmonella, Shigella, Salmonella typhi and Vibrio contaminate water. Human body contain these different types of bacteria. When human body excretes its waste product, then these are excreted via rectum and then pollute the air, pollute the water. Intestinal virus of animal that contaminate water by causing disease called diarrhea and enterovirus. Different viruses are also present, which also damage to the various water surfaces. Water is also transport microorganism by surface and parts of ships in oceans. Different parts of ships in ocean carry this water and then transfer it to the other part. During their journey around the globe, when they move from all over the world, they carry that polluted water and transfer it from one place to other place. One of these organisms is Vibrio cholera, causes life-threatening diarrhea in humans, which is transported. Alright, moving toward the other point, freshwater microbiology. Freshwater microbiology means, first of all, the freshwater is divided into 3 zones, 3 basic groups: littoral zone, limnatic zone, benthic zone. Littoral zone, limnatic zone and benthic zone. The variety of microorganisms live in freshwater. The region of water body near the shoreline is well lightened, shallow and warmer than the region. Photosynthetic bacteria and algae uses light as energy source and thrive and survive in this zone. Alright. The corners of, basically we can say, the corners of the freshwater area, like in streams, if we talk about stream, or we can talk about seawater, or we can talk about simple water standing, then in the margins, in the corners, that is littoral zone. Well lighted area where photosynthetic algae and bacteria is present. As the light is sunlight, then that sunlight is used by these algae and these bacteria, and they use them to prepare their energy. The second one is the limnatic zone. The second one is limnatic zone. Away from the shoreline is limnatic zone. Moving slightly away from the corners, there is limnatic zone. As the water deepens, the temperature becomes colder. The temperature slightly lowers and oxygen and light decreases. Purple and green sulfur anaerobic bacteria are dominant. Why anaerobic? Because there is less oxygen in this zone. So there is the presence of those bacteria which survive in the presence of, in the absence of oxygen. That are anaerobic bacteria. They will be present there. And which type of anaerobic is present? That is purple green sulfur bacteria. Alright. Photosynthetic bacteria are also present here because there is little light. The presence of light is favorable for the process of photosynthesis. The third one is the benthic zone. The bottom of the freshwater or bottom of the ocean, sea, where few microorganisms survives, because there is no light, there is no oxygen, there is no other proper condition where bacteria or virus could grow. Bacteria live without oxygen are present there. Bacteria that live without sunlight are present there. Now there is a condition change, not only anaerobic but also those which do not require any light. Only they will be present in the benthic zone, and these are basically methane producing bacteria that survive in this environment. Methane producing bacteria survive in this environment. You can see here in the diagram, the very interesting diagram, like the first one is littoral zone. You can see here different plants growing there, different lighted areas. You will see these margins on both sides. This lighted area, this is littoral zone, well lighted area. you can see here. Then moving slightly away from the shoreline, you see here limnatic zone, where less oxygen, where less sunlight. Okay. Then there is profundal zone between limnatic and benthic zone, where few may survive, like they may be anaerobic, they may be aerobic, they may be photosynthetic, very little oxygen, very little sunlight is present. Then in the land we have benthic zone. We have benthic zone, and in benthic zone there is no other, you can see here, but there you can see these fishes and animals, but in benthic zone there is no other species living there. Only methane producing bacteria survive there. There is no light, there is no oxygen, so bacteria were not present there. Then we will discuss about this saltwater microbiology. Salt loving bacteria basically present in this water, and these bacteria are called as halophilic bacteria. Halophilic bacteria. Archaebacteria is dominant form of life in the ocean. Archaebacteria is the dominant form of life in the ocean. If the bacteria are salt loving, then they are called halophiles, and these halophiles are present in the seawater. Basically, seawater is salty. If we study the microbial flora of water, then water supports the growth of beneficial bacteria. For example, certain yeast provide us with beer and bread. Similarly, some bacteria grows in contaminated water, help in digestion of the poison from the environment. You got it? Some yeast provide us beer and bread. Similarly, some bacteria that grow in contaminated water are helpful in digesting the poison from the water. If we have water which is contaminated, then some bacteria were grown there and they help to digest that poison from the water, and they are very, very dangerous. Diseases like microbes in water are those diseases which are caused by the microorganism in water are very life-threatening, like bacteria and viruses live in the intestine of human and animals. Found in the feces can contaminate the water and can be fatal after drinking. For example, E. coli, Salmonella, Shigella, Vibrio cholera, rotavirus, enterovirus, coccidiovirus. Alright, these are basically those which are present in water, and they are very, very dangerous to human health. They are very, very dangerous to human health. I hope you got this point, that microflora mean that environment we have where microorganisms are present. Okay, different bacteria, different viruses are present and living there and growing there. If that point is basically of human intestine or animal feces, then this contains various disastrous chemicals which are very dangerous to human health. If humans have that contaminated water, drink it. Microbes like protozoa, Giardia and Cystosporidium in drinking water causes prolonged diarrhea and fatal for impaired immune system. If we have protozoa like Giardia or Cystosporidium, then these Giardia and Cystosporidium in drinking water are very dangerous to human health. They can cause prolonged long-term diarrhea and fatal for impaired immune system. Those having very, very low immunity are very at the peak of their death if they have this type of infection. They have that water that contain microorganism protozoa. Life-threatening condition occurs. Many microbes like bacteria, protozoa, algae, fungi, tiny animals, etc. are important in food chain for life in water. For example, cyanobacteria convert sunlight into energy useful for health life, for water life. Algae is also used as important food for other life of water. Alright, many microorganisms like bacteria, protozoa, algae, fungi and tiny animals are important in the food chain of life, for life in water. There are many other microorganisms which are present in water and they need that protozoa, that virus, that fungi for their food chain. For example, cyanobacteria, which is also known as blue-green algae, it converts sunlight into energy using water life. It uses the microorganism present in the water and convert that sunlight into food. Algae is also used as important food for other life. Algae is eaten by many other microorganisms used as food for them. Alright, means sometimes microorganisms are very dangerous, but sometimes they are also dangerous in the way that they are helpful for the production of other microorganisms too. They are having relationship with them. There may be a symbiotic relationship between them, which causes them to grow by getting their specific nutrition. As you are seeing here, blue-green algae that converts sunlight into the energy useful for water life. Then microorganisms present in fresh water. As we have discussed earlier, different zones of water. The first one, the region of the water body near the shoreline, which is well-lighted area, a shallow area, warmer area having high temperature. Having high temperature. There, photosynthetic algae is present. Photosynthetic bacteria are present. They use that light and convert it into energy. Further moving away towards limnatic zone, photosynthetic microorganisms are also present here, but with the depends of water, light, temperature and oxygen decreases. So microbes require oxygen, don't survive there. Only anaerobic bacteria will be there. Bacteria are present, but why not three different types of bacteria? Some are aerobic, which need oxygen for their growth. Some are anaerobic. They do not require any oxygen for their growth. They may survive in the absence of oxygen. They may survive in the less oxygen. And some are those which survive in both conditions. They may survive in the presence of water. They may survive in the absence of water. Alright. So, in the limnatic zone, the microorganisms present are only anaerobic in nature. If they are bacteria, they are anaerobic, which do not require any oxygen for their survival. Then moving towards the freshwater, third zone, benthic zone, lowest zone. Only few microorganisms are present there because of the absence of sunlight and oxygen. And the temperature is colder, very, very low temperature. Few microorganisms survive there which do not require any oxygen, which do not require any sunlight for growth. As I told you about methane producing bacteria which are present in the lowest layer. Then microorganisms present in the salt water. Salt water presents different environment to the microbe. Different environment to the microorganism. Higher the concentration of salt, higher the pH. It will be more than 7. lower nutrients relative to fresh water are lethal to life. Means if salt is more in any water area, then we will say that there is less nutrient. But salt loving bacteria, only those bacteria that only need salt for their survival, are present there. And those microbes, for example Pseudomonas and Vibrioids, are also present there. Archaea is one of the dominant form of life zone in this area, in this salt water zone. The first one survival is halophilic bacteria. The second one survival is for the Vibrio cholera and Pseudomonas. The third one survival is for Archaebacteria, and these Archaebacteria, ancient bacteria, are very dominant species. They are very environmental specific species. Dinoflagellates, type of algae, also found in fresh salt water. The rapid growth and multiplication of dinoflagellates turn the water red. As the number of dinoflagellates increases, the color of water changes to red. This red tide depletes the nutrients and oxygen of water and causes the fishes to die. You can see here in the diagram red tide. Here salt water is present, and that salt water contains dinoflagellates. These dinoflagellates, when multiplicates, then they turn the water red, and they causes the absence of, they depletes the presence of nutrition there, and oxygen is not present there. Therefore the survival of the microorganisms is very difficult. Water is the transport of microorganism. How water is used? As we have discussed, air is our transport media. Water is also a good transport media. Water is ideal mode of transportation for microorganism. For example, water in the body of ships to stabilize the vessel and tank during voyages is now to be a main mode of transportation around the globe. Like Vibrio cholera causes diarrhea in human, and it is transported via ships from one area to the other area, and that water, when used by the humans as a drinking water, then this will cause severe diarrhea. The basic media for transporting microorganism, it is. Alright. Then moving toward the other topic, how we kill these bacteria. How we kill these bacteria. Bacteria killing treatment is done to have this type of remedy. Drinking water is usually treated to minimize the risk of microbial contamination. Drinking water is treated to minimize the risk of microbial contamination. Chlorine and chlorine derivative are used to kill the bacteria like E. coli in water. Chlorine treatment is done, which is very effective for killing E. coli bacteria, which is very larger in number present in that. So we have chlorine treatment for this purpose of remedy. Gases and radiations are used to kill the microbes in drinking water. For example, ozone gas and UV rays are used for killing the microorganisms present in the water, present in the drinking water especially. The microbes can also be physically excluded from water by filters. We can use the process of filtration, and we can have the separation of these microorganisms simply by filtration using different filter medias, different filter papers having different pore size. What is water quality testing? An important aspect of microbiology for drinking water is testing of water to ensure that it is safe to drink. It is safe to drink. It must be ensured. It must be confirmed that the water is safe for drinking. For that purpose, we have to do test which is called water quality test. Turbidity test of water gives an indication of the amount of suspended particles in the water. We will perform a test which is called turbidity test, in which we have a liquid which is unclear. That means it is turbid. That turbidity shows that there are particles suspended in the water. In this test, when light is passed through the sample water at 90 degree, the nephelometric turbidity unit is measured. A nephelometer is used to measure this. Instrument used to measure turbidity is measure the intensity of light scattered at 90 degree. So if the water's quality is deteriorating, action should be taken. If the angle is 90 degree or away from it, then we will say that there is some turbidity, and that turbidity shows that there is some contamination in the water. Nephelometric turbidity unit is measured in this way, and the apparatus used, device used is called nephelometer. Alright. If in measure of intensity of light, if the light is scattering at the angle of 90 degree, then we will say the quality is deteriorating. So many chemicals are present, so many pollutants are present which have contaminated the water. Alright, this is basically the water quality testing. Then we have microbiology of soil. The branch of microbiology which deals with the study of soil microorganism and their activities in the soil. The branch of microbiology which deals with the study of soil microorganism and their activities in the soil. Activities means different cycles they perform during their life cycle. Maybe nitrogen cycle, carbon cycle, oxygen cycle. Different cycles are carried out to have different nutrients present in the soil, and microorganisms undergo that metabolic processes in the soil. Then the study of these microorganisms basically that have different soil activities to enhance its fertility, then we can say this is the microbiology of soil. Soil can also be defined as organic and inorganic matter on the surface of earth. Any matter that is organic in nature, that is inorganic in nature, they both present on the surface upper layer of the earth, that provide a medium for plant to grow, the basic medium of the plant for growth. Alright. The plant grows because of soil. Every vegetable, every plant, smaller, larger, bigger, herb, shrub, trees, everything is possible due to the presence of soil. And the soil contains different organic matter and different inorganic matters. Agriculturally, the soil is the region which supports the plant life. How it supports the plant life? By providing mechanical support, strength. First of all, it provides strength to that plant to get itself being trapped into the soil, and nutrients are required for this growth. Two things are provided by soil to plant. The first one is the mechanical support and the other one is the nutrition. These two major things are provided to the plant by soil. It is the most biological interaction site. The most abundant, we can say that the mostly used biological interaction site. What are the basic components of soil? Admixtures of 5 major components. The soil is basically the mixture of 5 components: organic matter, mineral matter, or we can say inorganic matter, soil air, which also means soil gases, soil water, which also means soil liquid, and soil microorganisms. These 5 things are combined and soil is formed. Soil is not only gas, soil is not only liquid, soil is not only microorganism, it is not only organic matter, it is not only inorganic matter, it is the combination of all these 5 ingredients in different ratios. Alright? It is a combination of these 5 different major components, and these 5 components are combined in different percentages, different concentration, different ratios, and make out a soil, a good soil. Moving toward the mineral matter, as I told you, inorganic matter. It is derived from the bedrocks through decomposition. The process of decomposition, process of breakdown, disintegration is done. The weathering process is basically takes place. What is weathering? It is a mechanical and chemical process by which rocks are broken down into small pieces. Any mechanical process, any chemical process by which rocks are broken down into smaller pieces. Inorganic compounds include silicon, aluminium, iron, and other, maybe carbon, calcium, potassium, manganese, sodium, sulfur, phosphorus, etc. These are different inorganic points, inorganic chemicals: silicon, aluminium, iron, carbon, oxygen, potassium, sodium, sulfur, and phosphorus. Inorganic matter. Proportion of mineral less than half of total volume of soil. Less than 50% is inorganic matter. You can easily say less than 50%. Now organic matter. It is organic residue of plant and animal added in the soil. The waste which animal or plants added is organic in nature. It is not only providing the food for microorganism, but also provide energy for metabolic processes of microbes. Alright? Not only provide food, but also provide energy. And this organic matter is a potential, the most important source for nitrogen, for phosphorus, for sulfur, plant growth. These are basic needs for the growth of plant. Microbial decomposition of organic matter releases many unavailable nutrients to the soil. There are many nutrients which are not present, which are not available to the soil for growth. These are basically obtained to the soil by the process of decomposition of this soil by using microorganisms. Proportion of organic matter is from 3 to 6% of total volume of soil. Inorganic matter is less than 50%. Organic matter is 3 to 6% of total soil volume. Alright, done with it. Organic and inorganic matter. We can say 50% of the soil is due to the organic and inorganic matter, and from organic and inorganic, the major portion is of organic matter and the very light portion is of organic matter. Then soil water or liquid soil, you can say. The soil water comes from the rain, snow, dew, irrigation. These are basic reasons by which the soil get water. Microorganisms in soil require water for their metabolic activities. Obviously their metabolism require water, and that water present in the soil. Where it comes? It comes from rain, it may come from snow, it may come from dew, it may come from irrigation. Whenever we plant something in the soil, we have to provide them with water. We have to provide them with proper water, proper liquid. So that the microorganism present in the soil uses that water and start their metabolic processes and increase the fertility of soil. It affects the plant growth by its effect on the soil and organism. Percentage of soil water is 25%. Now if we calculate, then we have round about 30% left concentration. The 50%, or you can say 45 to 50% is inorganic, 3 to 6% is organic, and 25% volume is for soil water. In the diagram you can see here there is very less water, then there is somehow more water, then this is full of water. This is a very good part for the soil, very important for the soil activity. Then soil air. Soil volume is which is not occupied by soil particle like pore spaces are filled by soil water and partially with the soil air. You can see here in the diagram also there is some water and some spaces. This is soil air. This small air space contain different gases which is useful. For example, you can see here which is not occupied by soil particle like spaces which are filled by soil water and partially with the air. You can see here partially filled with air, partially filled with water. Before aeration, non-aeration, and after aeration. You can see here more growth. Soil, water and air are those components that make half of the soil volume. Means 50% is organic and inorganic matter. The other 50% is soil, water and air. Soil is lower in oxygen but higher in carbon dioxide as compared to atmosphere, because carbon dioxide is continuous process by microorganisms. As living systems are present, then they releases carbon dioxide all the time and oxygen level is very less there. That oxygen is present in only these spaces. These air spaces only contain oxygen which microorganism uses, and also present with water that microorganism uses. The oxygen from these two sources are used by microorganism, and in return carbon dioxide is produced in excess. Soil air comes from external atmosphere containing nitrogen, oxygen, carbon dioxide and water. All these are present in the soil air. Soil aeration plays an important role in the plant growth. Aeration means providing air to the soil. It is very important process. During the process of irrigation, we use this process. We provide air, we provide water, both, and both these contain oxygen which is used by the microorganism and the activities are performed. In return, carbon dioxide is continuously produced, which increases the level of carbon dioxide. Soil air contains 0.3 to 1% water, while our atmosphere air contains 0.03% water. The air is rich in carbon dioxide. So air in soil, or you can say soil air is rich in carbon dioxide as compared to oxygen. And if we compare soil air with the atmospheric air, then we will say that soil air contains more carbon dioxide as compared to atmospheric air carbon dioxide. In the atmospheric air, carbon dioxide level is 0.03%, while in soil air carbon dioxide level is about 0.3 to 1%. A very big difference between them. Then we have soil microbiology. as we have discussed about these inorganic matter present, organic present, soil water present, soil air present. Then microorganisms which are present in the soil are, it is excellent culture media for the growth of microorganisms. For other purposes, we need to prepare a culture media using different nutrition, using different chemicals, using different nitrogen source, carbon source, phosphorus source. But in soil, we do not need anything. We just need few things like water, oxygen, carbon dioxide to start the reaction of metabolism by microorganisms, and then soil plays its role as a culture media. It is dynamic living system because it contains various group of microbes like bacteria, like algae, like fungi, like actinomycetes, like protozoa, virus, but the bacteria are most numerous. Bacteria are most numerous. These microbes are less than 1% in total volume, and in upper portion of soil, microbial population is very high and decreases with depth. As we go more lower, then oxygen level decreases, water level decreases, sunlight decreases, and the survival of the microorganisms is on chance. Few microorganisms survives where oxygen is not present. Few are survived where both oxygen and light is not present. Then we will say that it is specific to the depth of the soil. Moving deeper causes decrease in the contamination. Then living organisms in soil are divided into two categories: soil flora and soil fauna. Flora means organisms that lives in specific place or environment, like bacteria, like fungi, like algae, actinomycetes. And soil fauna means a group of animals, not microorganism, the group of animals that are present in a particular region. For example, protozoa, nematode, earthworm, leeches, etc. You can see here percentage of various soil microorganisms. Bacteria, aerobic bacteria are 70%. Then moving towards slightly deeper, there is anaerobic bacteria 30%. Further going beneath the earth, actinomycetes are 13%. Fungi and molds are 3% only, and algae viruses are 0.2 to 0.8% only. This is a basic microorganism concentration. Alright. The most abundant are aerobic bacteria, then anaerobic, then actinomycetes, then fungi and molds, then algae, virus and protozoa, and in the end all these are combined and make up to 100% of the total volume of the soil. Alright. Then importance of microbiology of soil. Living organisms, plants and animals types are important component of soil. It plays a very important role in plant animal ecosystem. The scope of soil microbiology can be understood by studying various aspects, like soil is a living system. Soil is a living system. Soil microbes and plant growth. It is very important aspect. Soil microorganism and soil structure. Structure of soil is very important. In the study of soil microbiology, the structure of soil is very important. The presence of microorganism in soil is very important. The decomposition of organic matter is of great consideration. The formation of humus is of great aspect. Like humus, what is humus? It is plant soil fertilizer or rich soil by decaying matter. Basically, it is, you can say, a fertility enhancer for soil. Then geochemical cycling of element. Different elements like nitrogen, like carbon, like oxygen, like water, like ammonia. All these cycles are basically due to this soil. Soil microorganism as biocontrol agent. Microorganism present act as biocontrol agent. They control the pests, they control the insect attack. Soil microbes and seed germination. It is of great importance. As the fertility of soil increases, the certain germination of seed increases, which is then converted into a whole plant. And in the end, biological nitrogen fixation. You can see here different definitions like biochemical agent, biogeochemical agents, which is basically the study of connection between living organism and chemical elements. Biocontrol agents are the agents that are used to control the pests such as insect, mites, and plant disease causing harmful organisms. Nitrogen fixation is a process by which molecular nitrogen in the air is converted into ammonia. Atmospheric nitrogen is molecular dinitrogen, relatively non-reactive molecule that is metabolically useless to all but a few organisms useless. So this atmospheric nitrogen is converted into ammonia, and then microorganism uses this ammonia to convert it into urea and then use it in the formation of further nitrogen. Cycle continues. This is all for today's lecture, in which we have discussed about the microbiology of our environment, including air, water, and soil. Thank you so much. Stay safe, stay blessed.